Hi everyone, welcome back. Welcome to Medical Science. Thank you all for tuning in to our third video. We're going to be starting our new series today on hematology, and we're basically going to be talking about the formation of the platelet plug as well as the coagulative process. Let's start with a quick discussion on the difference between hemostasis versus thrombosis. So hemostasis is a physiologic process versus thrombosis being a pathologic process. A pathologic process that can sometimes occur without a state of injury, whereas hemostasis will typically be associated with an injured blood vessels to get the bleeding under control. So the first thing we want to ask ourselves here is what type of blood vessel is affected? So the main type of blood vessels are the small blood vessels that get affected, which can include the arterioles, the venules, as well as the capillaries. So what we're going to do now is we're going to schematically kind of show a regular blood vessel and sort of walk through the process of when there's a site of injury, how a platelet plug is going to form. So here's our blood vessel drawn over here, and now I'm going to draw a second blood vessel, and as you can see, I kind of point out with the circle that there is a site of injury at that blood vessel. We've endured some damage, and now we need our platelet plug to form, to begin forming. So the first step in that is going to be the process of vasoconstriction. Okay, so we're going to start out with a vasoconstrictive process. This process is mediated in a couple different ways, but basically the vasoconstriction process is just going to mean that the vessel is going to constrict. It's going to get smaller in diameter, and that's going to basically be to slow down the outflow of blood from the site of injury, to slow down the flow of blood at that particular site, and that happens through two different ways. There is a reflexive response, sort of like a reflex mechanically. When you touch like a hot plate, this is your vessels responding reflexively in a neurogenic mechanism, so your brain sort of just reacts to that. And then the other way is through derivatives in your body, substances that are produced called endothelin. And this is produced by your endothelial cells that are damaged, and that results in the vasoconstrictive process as well. So as I mentioned before, basically the decrease in the diameter of the blood vessel is going to lead to a decrease in the velocity of blood flow. This transient vasoconstrictive process essentially allows the platelets to begin sticking to the walls of the blood vessel to form our primary hemostatic plug, which is what we're going to discuss next. So in step 2, our story continues for the hemostatic process. Like we said in step 1, we had the transient vasoconstriction that occurred, and usually the flow is laminar, and by vasoconstricting, that laminar flow actually prevents the adhesion of cells to the walls of the blood vessels, so we're not constantly throwing up clots. In this case, we have that vasoconstrictive process so that this platelet plug can begin to form. So now that we've kind of initiated that cascade, we're going to sort of go into a deeper discussion on the steps that occur for this process to happen. So with regards to the formation of the platelet plug, essentially when the blood vessel is injured, vasoconstriction has occurred, we're sort of exposed to this underlying milieu of things, which consists of fibroblasts, smooth muscle cells, which here is abbreviated as SMC, subendothelial collagen, and tissue factor. And also, one of the main components that'll be exposed will be the subendothelial von Willebrand factor. Now, many of you may have heard of this. Von Willebrand factor is a protein. This protein will bind to platelets through a receptor that's present on platelets called the glycoprotein 1b receptor. That is a transmembrane receptor, and that will facilitate the binding of the platelet to the von Willebrand factor, which actually is bound to the subendothelial collagen, and this is kind of how the platelets start to stick to the outer wall and start to initiate that plug formation. The other thing to consider is the sources that this subendothelial von Willebrand factor comes from, and there's two of them. There is the endothelial cells themselves that will produce it, and then there's the platelets, which contain these Weibel-Palade bodies, and these are present in the platelets. So when von Willebrand factor, the reason why it's so important clinically is because there can be pathology associated with this factor. If there's too much of it, you can be in a state of thrombosis. If there's not enough of it, then you can have bleeding disorders. So, we're talking a lot about the platelet plug formation and sort of how sequentially this process occurs, but we don't want to lose sight of the fact that there's this cascade of events that's occurring concurrently. And in the previous slide, we talked about the role of subendothelial collagen and how it binds to von Willebrand factor, which therefore then binds to the platelets. But collagen has many roles here. One of its roles is binding to another factor and sort of activating it, and that is factor 12, which is also known as the Hageman factor. And this factor is involved in initiating the intrinsic pathway of coagulation. Now, coagulation comes in two different pathways. There is the intrinsic pathway, and there is the extrinsic pathway, which is facilitated by factor 7 and tissue factor. But here, collagen specifically works with factor 12 to facilitate the activation of the intrinsic pathway of coagulation, and all of this is occurring simultaneously to make a platelet plug become a stable platelet plug. But anyways, let's continue with our discussion in terms of platelets. So we are on step 3 now, and we are going to talk about platelet activation. So the two things that occur here are the conformational changes and a release of mediators, both contributing to platelet activation. In regards to the conformational shape changes, what we have to consider is that when that change in shape of the platelets is going to occur, that shape change is going to result in an increase in surface area. And if that surface area is increased, the binding capacity of the platelets is going to increase, and it's going to result in the exposure of a receptor known as glycoprotein 2b3a, which is very important in terms of platelet-to-platelet adhesion and further activation. In addition to that, there's going to be an exposure of negatively charged phospholipids, which are then going to be able to bind to platelets, further contributing to this activation process. In regards to the mediators that are released during the platelet activation process, once the conformational changes occur, the platelets will release substances such as ADP and thromboxane A2, which are going to have a multitude of effects and play a role in platelet activation. So now that we're on step 4, we're starting to talk about platelet aggregation. In the previous step, we mentioned that as far as platelet activation is concerned, there are two major processes, which are the conformational changes and the release of mediators. When it comes to the release of mediators, ADP is an important one. The release of ADP can result in the exposure of the receptor, which is the glycoprotein 2b3a receptor. This receptor is important because when it is not present or it is defective, it can result in a pathology known as Glanzmann thrombasthenia. In addition to that, ADP also serves as an important drug target when treating patients with clotting disorders. It functions as a drug target for a drug known as clopidogrel that many of you may be aware of. Next, moving forward, when we think about glycoprotein 2b3a, it actually promotes platelet aggregation by binding to a bivalent polypeptide known as fibrinogen, also known as factor 1. the binding of glycoprotein 2B, 3A to fibrinogen, which, like I said, is a bivalent polypeptide, allows platelets themselves to come together with one another. Steps of vessel injury, vasoconstriction, followed by platelet activation steps, aggregation steps, the platelets adhering to a damaged vessel wall and then further aggregating to one another. All of those steps have led to us forming the platelet plug, which is part of the primary hemostatic process. The important thing for us to keep in mind, however, is that the platelet plug that we have formed initially is unstable. This can be dislodged very easily. So we need to consider next is how are we going to go about stabilizing our platelet plug, and that's where secondary hemostasis comes to play. So now, as we can see here, we have our injured blood vessel, got the platelet plug like we mentioned before, and as far as the stabilization of the platelet plug is concerned, our main protein here is going to be fibrin. And this is really where now our discussion sort of shifts into talking about the clotting cascade, which, like I said before, this is all occurring concurrently. This is a simultaneous process, but we're talking about it individually for clarity. So let's go into our discussion about the clotting cascade. As I mentioned before, there's two different legs to this. There's the intrinsic as well as the extrinsic leg of the clotting cascade. And the important thing is the factors that are involved in it. So like I said before, the intrinsic cascade starts from factor 12 and starts to work down, and then the extrinsic cascade will start at factor 7 and with tissue factor. They do come down together into a common pathway, but let's start from the top here. So now we're starting to see this cascade sort of come together. So let's just give a quick overview of it. As you can see the things coming together with the intrinsic and extrinsic cascade, you can kind of follow along with the numbers that are represented as Roman numerals here. But I'll kind of detail things out. So essentially this cascade involves three different things. It requires an enzyme, a substrate, and a cofactor. So the cofactors here are going to be the calcium, and then the enzymes are actually the activated forms of our factors. And then we have the intrinsic, the extrinsic, and a common pathway. The intrinsic pathway has to do with contact with the surface. So we talked before about the exposure of subendothelial collagen and how that collagen plays a role in binding factor 12 and activating it to 12A. So that's where the intrinsic pathway comes into play. The extrinsic pathway, on the other hand, has to do with trauma, tissue trauma. That trauma itself is going to release a substance called tissue factor, and that will result in the activation of factor 7, to factor 7A, and then ultimately they come down and meet at the common pathway, and the common pathway begins at factor 10, which then gets activated to 10A, and in combination with factor 5A activates thrombin, which is factor 2, into 2A, which then goes on and plays various different roles. It plays a role in negative feedback of this entire clotting cascade, but also goes on to activate fibrinogen, which is factor 1, into factor 1A, which is fibrin, and then we form a stable cross-link clot by further activating factor 13 into 13A. So in the next steps, we'll talk a little bit more about the roles of thrombin. So as our story continues in the coagulation cascade and this secondary hemostatic plug, now we start to change our discussion into the role of thrombin. And thrombin is actually a critical factor in this whole process. Thrombin, like we said before, is factor 2, and it plays several roles when it comes to this whole process. In addition to its role in converting the fibrinogen, which is the inactive form, into fibrin and forming a cross-linked platelet plug, it's also, like I said, it's involved in positive and negative feedback mechanisms, which allows it to function as a platelet activator. It induces platelet activation, and that's why I mentioned earlier that this is a concurrent process. This is all happening at the same time, and the way it does that is through a protease activating receptor, which is a type of G protein coupled receptor, and it binds to that and induces further platelet activation. It also uses that same receptor for its pro-inflammatory effects because those receptors are also expressed on different inflammatory cell types, and so that allows it to contribute to that role. The strange one here is going to be the anticoagulation effects of thrombin itself, and that actually has to do with its interaction with a normal endothelial surface. So there's two really important things to consider here. That once we form this clot, that clot has stabilized. Well, we talked about how there's pathology associated with clot formation, aberrant clot formation. So most importantly, once the repair process occurred and initiated, the clot must be able to dissolve. And also, it's important that the site where we have the damage is the only place where our clot is going to remain. So it's important that the clot remains confined to the site of injury. If it's starting to form on normal endothelial surfaces and other places in the body, that's when we've got a pathologic issue at hand, and that's when problems can occur. So it's important that things must remain confined. And uniquely enough, there's actually various different mechanisms the way our body is able to do that. One of the main ones would be the fibrinolytic cascade, but other factors would be blood kind of flowing through the site of injury and washing out the coagulation factors. Another would be that since we require these negatively charged surfaces that are provided by platelets and platelets that are activated, that's not going to be present, so that's another way. And so those are some of the ways that this thing can continue to be confined to the site of injury. So in this previous discussion, we've been talking a lot about how the clot needs to remain confined to the site of injury, and I mentioned how one of the main ways to do that is through the process of fibrinolysis. So it's important that we mention that one of the main factors that's involved in fibrinolysis is a factor known as TPA. And you may have heard of this clinically being administered, but also this factor is called the tissue plasminogen activator, and it's exactly what that is. It activates plasminogen into plasmin. Now, plasmin is a factor. It's a mediator that's involved in the breakdown of that fibrin-formed clot, fibrin-stabilized clot. This tissue plasminogen activator is made in the endothelium, and it breaks down. The activated plasmin is going to break down the fibrin-stabilized clot, and the breakdown products, one of those breakdown products is known as a D-dimer, and that actually holds clinical significance when we're looking at patients such as with DICs, where we might notice elevated D-dimer levels. It sort of has a predictive function in the type of pathology that they may be dealing with. So fibrinolysis is a very complex process, but to simplify it, we have TPA. TPA takes plasminogen, converts it to plasmin, and plasmin then starts to break down our fibrin-stabilized clot. So here you see kind of a blood vessel. You see one sort of cut section of a blood vessel, and you see these various endothelial cells that are lining that blood vessel. And the reason why I bring this up is because this is another sort of another cell type that's really heavily involved in the regulatory process when it comes to the coagulation cascade. So first and foremost, we'll talk about the role of thrombomodulin that is present. Thrombomodulin, like I said, the thrombin itself can have pro as well as anticoagulant effects, and this is where we're going to see thrombin's anticoagulant effect, in that it binds to thrombomodulin on a normal endothelial surface, and when it binds to a normal endothelial surface, it's going to be able to activate protein C using protein S as our cofactor and become activated protein C. So thrombin now is functioning as an anticoagulant, and that is going to result in its inactivation of factor 5A and factor 8A, which we saw previously had roles in the coagulation cascade, in the intrinsic as well as the common pathway of coagulation. So this is how thrombin actually sort of negatively feeds back on itself. And there's also the tissue factor pathway inhibitor, which inactivates the factor 7A complex. So as you can see, thrombin is kind of feeding back into the cycle and playing a role in controlling both the intrinsic and the extrinsic aspects of the coagulation cascade, and that's how it negatively feeds back on itself using the endothelium. So, as our discussion has been going, the endothelium is obviously very, very important when it comes to controlling the coagulation cascade, whether that be playing an inhibitory role in terms of platelets using PGI2 prostacyclin, or adenine diphosphatase in terms of breaking down ADP, or nitric oxide in terms of having a vasodilatory effect on the blood vessels. In addition to that, we talked about the role of TPA having a role in fibrinolysis, and lastly, there is the heparin-like molecules, which are going to bind to a protein called antithrombin 3, and antithrombin 3 can then actually inactivate thrombin as well as factors 9, 10, 11, and 12, and this is how the endothelium has a regulatory effect. So again, everyone, thanks so much for listening in on this video. I know there was a lot of information presented. If there's any clarifications that you would like, please leave some comments below. I'd be happy to touch base on it, and more videos to come in the future. Thanks so much. So if you would like to subscribe to our channel, go ahead and hit the link on the left side, or if you would like to check out one of the older videos in the cardiovascular series, go ahead and hit the link on the right. Thanks again.